is about application of enzymes from extreme files. First, I explain about extreme files, and then organic solvent-tolerant enzymes from organic solvent-tolerant microorganisms. Finally, I will talk about phasous-degrading enzyme from alkalophile. This work was done by Professor Inoue's group. First, I explain about extreme files very briefly. Moderate conditions, such as 25 degrees or pH 7, these conditions are ideal conditions for humans, but below 10 degrees or more than 60 degrees, low or high pH, high salt concentration. These conditions are extreme conditions for humans and other organisms. But some microorganisms are found in these extreme conditions. They are tolerant of extreme conditions. In many cases, they require the extreme in order to grow. These microorganisms have been referred to as extremophiles. Extremophiles are found in various habitats such as hot spring, deep sea, in ice, like this. This table shows industrial application of enzyme isolated from extremophiles. Enzymes from extremophiles are expected as biocatalysts in extreme conditions. For example, DNA polymerase from thermophile in use for polymerase chain reaction and proteinase and cellulase from alkalophile used as detergent enzymes are famous examples of enzymes from extremophiles. An organic solvent-tolerant bacterium was first reported by Professor Inoue and Horikoshi. They discovered a tolerant bacterium, Pseudomonas fuchsia IH2000. This strain can grow in the presence of 15% toluene, like this picture. This table shows organic solvent tolerance of several microorganisms. Growth of microorganisms in the presence of organic solvents was examined. These are solvent tested. Log PO value is one of the physiochemical parameters which shows polarity of some compound. Professor Inoue and Horikoshi proposed log PO value is inversely correlated with the toxicity of organic solvent. So an organic solvent with lower log PO value is more toxic than a solvent with higher log PO value. The tolerance level of each microorganism are different among genera, strain, species, strain, bacteria. IH2000 can grow in the presence of various organic solvents as compared with other microorganisms. Now we are trying to construct a biosensor for monitoring cholesterol by the use of WK cholesterol oxidase with Professor Okuma. He is a specialist in electrochemistry. By adding carbon nanotube to the reaction area of the electrode, we can construct a high-sensitive biosensor because carbon nanotube promotes the electron transfer. I think WK cholesterol oxidase is useful for this application. Finally, I will talk about phasous-degrading enzyme from alkalophile. This work was done by Professor Inoue's group. So large amounts of waste phasers are generated as byproduct in poultry industry. Most such phasers are simply incinerated. So phasers are composed mainly of a protein. So efficient method of waste phasers degradation is desired for recovering amino acid. So they screened phasor degrading bacteria with alkaline medium containing chicken feathers powder. So this picture shows halo formation by phasor degrading bacteria on one agar medium containing feathers powder. Strain FA3001 showed the highest activity. So this picture shows this integration of native chicken feathers by culture filtrate of the strain at different temperatures. Native chicken feathers degrading activities were found at 30 degrees to 60 degrees. This figure shows effects of pH and temperature on the proteinase activity. Optimum activity at 30 degrees or 40 degrees, optimum activity was found at near pH 12. Optimal activities were shifted to acidic pH at higher temperature. At 60 degrees, optimum, the highest activity was observed at pH 9 to 10. This enzyme was first reported as feather-degrading proteinase, which shows high activity at alkaline pH. So they are now studying, as Professor Maekawa talked, so they are studying activation of proteinase by the addition of CMT. When CMT was added to the reaction solution of proteinase, the activity was increased